Hi, everyone. I'm actually working to get the closed captioning. I'm not sure why it is not appearing as it should be. I apologize for the inconvenience. I am working on it. Sarah, if you want to get started.
Yeah, sure. Tiffany is going to get us started, so I will go ahead and pass it to her for our kickoff.
Tiffany, are you there? Oh.
Yes. Can you hear me now?
Yes.
The tagline of 2022. Hey everyone, my name is Tiffany Sammons. I am the Ohio State Ambassador. Currently, however, I will be stepping down after this event, and Sarah will be filling my shoes and leaving Ohio in great hands here. Just a couple of announcements really quickly. Give me one moment here. This is our third annual Rare Disease Day in conjunction with the Connor B. Judge Foundation. And I will now be working for NORD directly, so you haven't lost me completely yet. I want to say that if you're not speaking, we ask that you do please mute. And we will also be giving away 2 door prizes today for all people that are registered. And I want to also just put the disclaimer out there that although there are several doctors on our line today, this is not a platform for any medical advice, and we do ask that you turn to your medical healthcare providers for that. I want to turn this over to Sarah so she can introduce herself and tell you more about the Ohio Rare Action Network.
Hi, everyone. I'm Sarah Sharp. I am a rare disease patient and advocate, and I'm also Ohio's State Ambassador for the Rare Action Network. The Rare Action Network is powered by NORD, or the National Organization for Rare Disorders, and is a network of advocates and organizations that have been empowered by education and resources from NORD to advocate for the rare disease community. We have a fabulous agenda today that includes both clinician, patient, and family member and parent perspectives. And we are so excited to have you joining us. This year, let's see, we can jump to the next slide, Tiffany, if you want. This year, we are going to be moving towards more community engagement, and there are going to be more opportunities for volunteering with Ohio's Rare Action Network, including I'll be looking for a co-ambassador eventually. So please, if you would like to get involved and volunteer with us, contact me. My information will be provided in the chat as well as at the end of this presentation. I'm looking forward to kicking things off by sending it over to Tiffany and Randy in just a moment to discuss the Ohio Rare Disease Advisory Council. But before that, I just wanted to give you a quick snapshot of our Ohio State report card. This is one way that NORD measures where we stand from a policy perspective. And we will also drop in the chat the link for the interactive state report card if you want more information about each of these areas and how you can help and advocate in your community. With that, I think I will give it to Tiffany and Randy.
Randy, I'm going to let you take the lead on this one.
Sure thing. Thank you, Tiffany, and thank you, Sarah. really glad to be here as I am a rare mom and former state representative in Ohio. And while I was serving, I was honored to draft and pass the Rare Disease Advisory Council for the state of Ohio. So I'm just going to give you a quick overview of what the council is and where we are and call to action. So the purpose of the Ohio's Rare Disease Advisory Council is to advise the General Assembly regarding research, diagnosis, treatment efforts in Ohio for patients with rare diseases. The original bill had a space for 25 members. We wrote our bill to have 5 patients and organizations. So that's 3 patients, 2 organizations that are represented here in Ohio. There's 2 spaces for research, 3 providers, 3 from the administration. So that's the Department of Medicaid, Department of Health, and Department of Insurance. There's a space for 5 associations, including the hospital, biopharma, health commissioners, and insurance. And then there's 7 elected officials, including the governor's office, House and Senate members. After that was put out there, there was a lot of interest in being on the Rare Disease Advisory Council, so much so that the Senate president introduced an amendment to our budget language this year to include 6 more appointments. Those can be anybody. We were lucky enough to get a couple more parents on in those 6 spaces, so we're really excited about that. So the main thing that we will get out of the Rare Disease Advisory Council is every 2 years, there will be a report that's put out of the incidence of rare disorders in the state of Ohio. I'm really excited about that because I think that that will drive innovation and technology to come to Ohio and help with our treatment efforts here. And then lastly, our big call to action is we have not yet met. We have potentially, we're supposed to meet this month or next month, sorry, March. So hopefully in March, we will have our first meeting, but the Speaker of the House has not yet appointed his members. So if you could contact Speaker Cupp's office and just ask for the appointments out of the Speaker's office, that would be great. We would love to have a full 31 members on our first meeting. So hopefully the speaker will appoint his appointees. If you have interest in being a part of the Rare Disease Advisory Council, the way the bill was written was that every 2 years, the governor makes the appointments to those 25 members. And nobody can serve more than 4 times. So if you are interested in being appointed, appointments will go out at the beginning of next year now. And we, Tiffany and I have talked about it. We are going to definitely have a way that families can be involved. So you can contact me or Tiffany, and we'll make sure that we have a coalition that will be formed to make sure that everything that's happening at the Rare Disease Advisory Council, families and associations and nonprofits, foundations that are operating here in the state of Ohio know what's going on with the council. So thank you so much for your time, and thank you to everybody on this call that helped pass that bill. We really appreciate your efforts.
Thank you, Randy. I just want to let everybody know, if you have any questions, drop them in the chat and we'll be sure to get those answered. For anybody that might not get their question answered, we will follow up with you after. So just drop those in the chat if you would. And next, I'd like to introduce Mitchell Drum. Mitch, I believe you're muted. If you could unmute your line, please.
Can you hear me now? Yeah, okay. Sorry about that. Yeah, so first of all, thank you all for joining us today. And I was given the charge to give you a little overview of where things are in the field right now, in the particularly for genetic disorders. And let me start by reminding you that, across your chromosomes, you've got about 20,000 different genes spread across there, and any one of them can, in theory, cause a genetic disease. And many of these you're all quite well aware of. I just threw a couple examples on here: sickle cell anemia, where you're affecting the blood and the hemoglobin; cystic fibrosis, where my history comes from, it affects the lungs, the GI systems; neurological disorders like Tay-Sachs; and metabolic disorders like phenylketonuria. But in addition to just those couple of examples, there's somewhere estimated between 7,000 and 10,000 other genetic disorders. And they're all incredibly rare. I don't need to tell you that, right? So 1 in 50,000, 1 in 100,000, things like that. But when you start putting them together as a group, you're now closer to 7,000, 10,000 different disorders that together bring the incidence somewhere around 1 in 20, some estimates even as high as 1 in 10 individuals will have a genetic disorder. So because of their rareness, it's hard to get things to move forward on any one disease. But when you think of them as a group, we have power in numbers. And it's organizations like NORD that allow that kind of progress to be made. So they are essential. So anyway, I thank them for including us today. This is really wonderful. So I have to do a little bit of Bio 101 to sort of set the stage for this. But if you think back just a few years ago, for most of these disorders, the only option was symptomatic therapy. So you know, if there's pain, inflammation, infection, whatever it is, that's where we were limited in treating things. But these genetic disorders, they start back at the chromosomal level. They're causes in your DNA. And the DNA is that instruction manual that then makes a copy of those genes in an RNA molecule. That RNA molecule gets processed into a thing called mRNA, and you've been hearing a lot about that in the news lately. But the mRNA is then instructs the cell to make a protein. And that's the guy that does the business. That's where the problems are for these things. But even though here's where it all originates, we treat it downstream typically. But over the past decade, there have been a lot of really impressive advancements in our understanding of the science so that we can now start to get closer to the cause of the disorder to treat it. And that's where we want to try to give you some snippets of some advances that have been happening more recently. So going one step back from the symptoms to the protein, most of us are familiar with what's called protein replacement therapy. So diabetes is probably the best example, where folks don't have enough insulin, so they can inject it right into their body and be treated from that. But a more recent example that has come up is in the development of pharmaceuticals that will act on the protein that's causing the disease, right? You get a headache, you take ibuprofen or Tylenol for the pain and stuff, but that doesn't really get to the source of it. Now we can have drugs that work on the inciting factor. And I think the next speaker may talk a little bit more about this, but the way pharmacology is now evolving is we can now test lots of drugs at the same time. And I make the little battleship analogy here that we don't test one chemical at a time to see if something will have an effect, but rather we can test tens of thousands of different drugs and see what works. And then once you see something that has a property you like, you can go in then and try to figure out what the good parts and the bad parts are of that chemical, and then get a chemist involved to try to convert that then to something that can be a drug. And I use the cystic fibrosis example because I think that's really one that's really illustrates how well this has worked. There are now there's a drug formulation here called Trikafta. It's a combination of 3 different chemicals that helps the CF protein fold into the way it's supposed to be so it can work, and then helps activate it once it folds correctly. And for many patients, this has been a life-changing event. So it's shown that this type of process is possible. So another example of getting at the taking one step back to the RNA level, is a disorder called spinal muscular atrophy. So spinal muscular atrophy causes severe neurological problems. So babies that are born with this will quickly develop paralysis and inability to move. And what was found out was that we know what gene causes that disorder, the SMN1 gene, but when it's got a mutation that prevents it from working, some clever folks figured out that, well, our genome has another gene that's very similar to that, but just it doesn't make much of it. So we can go in with a small piece of DNA called an oligonucleotide and trick the cell into using that other gene instead of the one that's mutated. And that will then replace functionally the mutant gene. And these kids have shown, the ones for whom this works, has shown that they can go from completely incapacitated to walking. And it's had an immense, again, another proof of concept of the kinds of things that we can do. And we take one more step back now to get to the DNA level. So we can treat at the protein level, we can treat at the RNA level, and we can treat it, well, we hope we can treat at the gene level. And there are a couple of examples of that from the typical rare genetic diseases, but also these technologies are being used in cancer as well. And then we'll touch on what I think is the newest of these technologies, and that is what's called genome editing, or we can actually get in and try to fix the gene itself. So here's one example of the gene therapy, and I'm going to make a distinction between what I call gene therapy and gene editing. Gene therapy meaning you're putting in a version of the gene that's not working, versus gene editing where you're actually fixing the gene, okay? So from the gene therapy standpoint, a target that has been looked at is our disorders of the eye, so things that will cause blindness. So this one here called Leber congenital amaurosis is a disease that can be caused by several types of genes and mutations in several different genes, but one in particular is the RPE65 gene. So it's there in the retina, and one of the things, properties of the eye that made this a place to test is, one, it's small, so you don't have to get a lot of stuff in there, but the other one is it's what we call immune privilege, so that you don't have to worry about the immune system going out and actually attacking what you're putting in there as a therapeutic. So what the technology allowed was to put a copy of this gene into a virus. So that pretty little blue and purple thing on there is a virus called adeno-associated virus. You put the gene in, you actually physically take a needle, you inject that into the eye, and it will create the protein that is defective in this disease and treat the disorder. A similar technology has been applied to some cancers now. And the idea here is, if you're aware of most cancer treatments, they go after to try to kill the cancer cells by using things that will target dividing cells. Tumors are dividing cells, so anything that would attack those cells would be a potential therapeutic. So we use chemicals that can be toxic, we can use radiation, but what came out was sort of the magic bullet approach, saying maybe we can use a person's own immune system to go after those cancer cells. So the process here is to take out one of your immune types of cells called T cells and put into them a gene that will target those immune cells to the cancer. So you take the cells out, put in the gene to target the cancer cells, put the cells back in, and they will then go after the tumors. Those are called CAR T cells, first chimeric antigen receptor T cells. So these are all things that are actually working and having a dramatic impact in many cases. And then the last part I wanted to touch on was what I'm sure all of you have been reading in the news for genetic disease, this stuff called CRISPR-Cas. Okay? So CRISPR-Cas, and not to be get into the weeds too much, so try to make an analogy here. This is an enzyme that we know will cut DNA, but it has a wonderful feature that throughout your 3 billion bases of DNA across your chromosomes, it can be targeted to a very specific region. So it doesn't go in and indiscriminately break up your chromosomes and cause more problems than it solves, but rather our chromosomes are barcoded. And so we can get this enzyme, this Cas9 enzyme, to go to a specific place. So imagine you can get it to target a mutation that causes the disease. It breaks the DNA. And then the cell, if there's a break in the DNA, the cell will repair that. And we can trick the cell into repairing it and getting rid of the mutation and putting in a normal sequence in the DNA to try to fix it. So there are a lot of different adjuncts that have been put into this process. So we won't go into all those, but they all rely on the fact that this can target a specific gene. And that was something that was missing from our technology toolbox until about 2013. So when CRISPR-Cas first finally came out, it really opened up a lot of possibilities. So we know we can do it. So now we're at the engineering stage for these kinds of therapies. We know we can do it. How do we do it in the right place? And how do we do it in enough cells that it will make a difference for an individual? And so you can understand that if I were working on, say, Duchenne muscular dystrophy, I need to get into a muscle cell. But if I'm working on a neurological disorder, I need to get into the brains, into the neurons, and so on and so forth. So the way we approach this then has to have some specificity for the disease. And I'll close with some of the other technologies. There are a lot of folks working on how to manipulate the DNA, but we also have to figure out how to know if it's working. And so one of the things, I'll start at the bottom here, one of the things we're doing is trying to develop ways that are non-invasive to actually see if this is working. So we have scientists here at our place who work with MRIs. And as an example, we went to them with our cystic fibrosis problems and we said, hey, we've got these young kids. How do we assess whether we fixed their lung function? And anybody who has a child knows you're not going to get a 2-year-old to sit still for 45 minutes when the MRI machine is going thunk, 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 thunk. So they were able to reprogram their scanners to do a 5-second scan and get enough information from that scan that they can see where the disease and the problems are in the lungs. We also do a lot of work with animal models. And this will sound perhaps a little Frankensteinish, but we've been able to put a gene from a lightning bug, the firefly, into mice and hook it up to a disease-causing gene. So if we can fix the gene, the mouse will light up. So all we have to do then is put the mouse to sleep for a little bit, try the therapy and see if they light up. And what you're seeing there on the right is what the machine looks like if we get them to light up. So the blues, the purples and things, that's showing where the mice have lit up. So that's it. I understand a very brief overview of these things, and I'm sorry we didn't get to meet this year in person to talk about these in face to face. But I want to close by saying that all the things that we and others are doing cannot happen without support from different groups. And we're, like I said, thrilled to be able to interface with NORD, but we also want to acknowledge that we started a system here at Case Western Reserve about 5-6 years ago called the Research Institute for Children's Health. And because of the successes we had in cystic fibrosis, we wanted to apply that process to as many other rare diseases as we could. So we set this up as an infrastructure, and we've been able to get started on about 24 different diseases as a consequence of that. And I'm going to use that as my segue because one of the folks that I've gotten to know very well through this process is my colleague, Paul Teasar. And Paul will tell you about the work that he's doing in disorders of the nervous system that affect myelination. So Paul has been a leader, a world leader in both the neurological disorders, but also in developing technologies in what are called induced pluripotent stem cells. So I'll stop there and hand it over to you, Paul.
Thanks very much, Mitch. It's really an honor to follow you. Mitch is an extremely modest guy, but for those in the audience who don't know, he's really over the course of his career been, a world leader in both the understanding the underlying cause of cystic fibrosis and really driving new therapeutics forward. So both from the scientific side and from the rare disease community side, Mitch, we all thank you for everything that you've done to bring cystic fibrosis to a point where there are life-changing therapeutics available to patients, and we hope that we can mimic, those types of efforts for other rare diseases. And this is one of the inspirations for a lot of the work that's ongoing in my own laboratory. All right, so I'm going to share my screen now. Look good for everyone? Terrific. All right, so thanks to Nord and the Conor B. Judge Foundation and all of the other organizers have put a lot of effort into today's virtual, hopefully next year not virtual, rare disease day presentation. And so I'd like to just share some of the research from my own laboratory today at Case Western Reserve University in the School of Medicine, and we won't get deep into the science, but what I hope that you'll take away from today is that it's really the collaboration of patients, patient advocates, scientists, and clinicians and pharmaceutical companies that come together to really advance science forward, from the lab into testing for patients. And so this is really the ultimate goal of my own laboratory, is to take the science that we drive forward in the laboratory from a discovery phase and advance it when appropriate safely into patients. And so, as Mitch mentioned, our lab is really a stem cell biology lab. We build cellular tools that allow us in a petri dish to have access to the cells that are normally locked within inside our brain. We can use those cells in a petri dish to then understand the underlying cause of neurological disease and to then discover and develop new medicines to treat those disorders. And so in my lab, we work on neurological disorders. Of course, everyone knows that nerve cells are the information superhighway of the brain. There's 100 billion of these nerve cells in the human brain. And we think about these nerve cells as a wire that transmit electrical signals that allow us to move, walk, talk, think, breathe, everything that we do on a daily basis. I think what's less appreciated is that these nerve cells are surrounded by an insulating coating, which is called myelin. Myelin is produced by a specialized cell type in the brain called oligodendrocytes, but you can think about this myelin coating as a protection around the nerve cell, but it also is required for the electrical signals to transmit along this nerve cell at the appropriate speeds. Right? And so what happens if you don't have myelin is it leads to disease. And so when you have dysfunction of the cells that make myelin or the myelin itself, this leads to a variety of neurological disorders, including things, well-known disease like multiple sclerosis, as well as disorders like neuromyelitis optica, which the Conor B. Judge Foundation has very kindly supported research in my lab focused on NMO. I'm going to talk today about a rare genetic disorder called Pelizaeus-Merzbacher disease that we work on in my laboratory. But there are a host of other diseases where there is myelin dysfunction involved. And so overall, the central theme and focus of my laboratory is to understand why this loss of myelin causes various different neurological disorders. And then more broadly is to develop therapeutics that can regenerate this protective myelin coating around nerve cells across a spectrum of neurological disease. So we've taken this framework and over the last 10 years, we've really pushed forward initially on thinking about how we could regenerate myelin in multiple sclerosis. And so we've built cellular tools that allow us to understand what's going wrong in the brain. We've understood when and where we need to regenerate this myelin coating, and we've developed new potential medicines. This program has now spun out into a biotechnology company that I co-founded here in Cleveland with my colleague Drew Adams, called PMD Therapeutics, which is now partnered with Genentech to advance these therapies into patients. But the story I'd like to tell you today is how we've used the same framework for a rare genetic myelin disorder called Pelizaeus-Merzbacher disease. And I think the most hopefully inspiring part of this story is this research progress started with these families right here. So this is the PMD Foundation. It's all family run. It's a small advocacy foundation where for patients and family members. They had a research grant back when I was a young new assistant professor, a small research grant available. And we felt in my lab that we were working on multiple sclerosis and that some of our technologies could apply to this rare genetic myelin disorder. And so we applied for the grant and we got it. And so that was day one of us working on PMD. And so, and what you'll see today is that we've really taken that and pushed that initial seed grant forward to lots of external funding, as well as to a potential therapy moving forward into clinical testing. And so just sort of a story that, for the patients and families and advocacy groups, that you really can make a difference. And we would not be working on this rare disease without this initial seed grant. And so what is PMD? So this is caused by mutations in a gene called PLP1. This gene is on the X chromosome. And so as a result, this disease affects young boys. It's characterized by dysfunction of the cells that create myelin and loss of myelin itself, that protective coating around your nerve cells. A host of cognitive and motor symptoms, and also there are early mortality for this disease. In particular, in the severe conatal form of this disease, kids succumb to the disorder in their very early years. And I think for us is that there were, at the time, what was really challenging was that there were no therapeutics available. All right. All right, so I won't go through this in detail, but this is also, even though it's a rare disease caused by mutations in a single gene, it's quite complicated in a sense, is that there's a spectrum of patients. The conatal severe form succumb to the disease very early in life. There's a classic form of the disease where the patients have less severe symptoms, but still quite impairing, and these patients live a bit longer into their 20s, 30s, and 40s typically. And so what did we do? Well, I told you that my lab is a stem cell biology lab, and we can take cells from patients and we can put them in a petri dish in a way that allows us to mimic what's going on in the brain. And so I'm just going to show you a video here, and I hope it plays. But what you'll see on the left is cells from a healthy patient, and on the right, cells from a patient with Pelizaeus-Merzbacher disease. And these are cells growing in a petri dish. What you'll see is on the left, the healthy cells are happy, they're moving around. They were going to extend out all of these little fingers and arms, which are little myelin projections, happily in the dish. But on the right, you'll see that these cells, for the most part, from the disease patient, they start to do this, but then they die. You see all these little white circles here. So what we've been able to do is, by really watching cells that are normally locked away in a patient's brain, but we can now watch them in real time and understand them in real time in the laboratory, we can really define when they die, why these cells die, what's going on, what's the dysfunction. And so this gives us a new window into understanding and developing how we might treat this disease, at what time we might treat this disease, and what therapeutics we might go after. And so after a number of years and a number of publications and testing and screening thousands of different approaches to fix this, this culminated in a paper that we published about 18 months in the premier scientific journal Nature, which we showed for the first time that we could have a dramatic impact on this disease in mice. And I'll show you the data here in a second, where we developed a new therapeutic approach to treat this disease. And so on the left here, so we took our therapeutics from the Petri dish now into mice. So we're using a mouse that has the same mutations as patients for Pelizaeus-Merzbacher disease. So it mimics that disease in patients. I'm showing you a section of brain tissue from a healthy mouse on the left here. Green staining is myelin, that's that protective coating. So there's lots of green, that's normal. In the PMD mouse here on the bottom, you can see there's very little green. So that's not good. That's why these mice develop disease and die quite early. But when we treat these mice with the therapy that we described in that paper, you can see that it restores all the green staining. So it restores the myelination. And excitingly, this is at 3 weeks of age. But if you look later at 6 months of age, here's the healthy mouse brain at 6 months of age, green staining, lots of myelin. Here's the PMD mouse treated with our therapy, basically equivalent to wild type levels. So we've restored the myelination in these brains. The PMD mice that are untreated, those mice all die at 3 weeks of age. And so this therapeutic not only restores myelin, but it extends the lifespan of these animals. I don't know if this video will play or not, but if it does, you can see the mouse on the right has PMD. It has this very severe attention tremor and ataxia, and the mouse on the left was treated with our therapeutic and is indistinguishable from wild type animals, full motor function. And so the most exciting thing for us was that this therapeutic that we developed, which is an antisense oligonucleotide, and so it targets the RNA, if you remember back to Mitch's talk, and then it prevents a toxic protein from being formed. This is a graph, this is a survival curve. And so the PMD mice with disease all die at 3 weeks of age. When we treat those mice, this dashed gray line here, they live out 12, 16, 18, 20 months of age. So these mice live now a full lifespan, which is pretty remarkable, right? We're not extending life by a couple of days or a couple of weeks in these animals, but we've provided a full lifespan extension. Here's one of our old PMD guys, fat and happy and grumpy and just moving around at 15 months of age. And so this was a pretty dramatic results. We received a lot of coverage in various press for this study that we published in Nature, and I'm very excited to report that last month in January we announced an agreement. Case Western Reserve University announced an agreement with Ionis Pharmaceuticals, a major pharmaceutical company in California, and the developer, along with Biogen, of the drug that Mitch mentioned for spinal muscular atrophy, Spinraza, to advance this therapy into clinical testing. And so I don't normally like to quote myself, but I had a quote in this press release that really is what I wanted to end this presentation on. So I just put it here. And for me, you know, as a PhD scientist, not a clinician, the ability to see the science move from the laboratory, you know, from a petri dish initially into mice and advance into patients is really the most gratifying thing that I think a scientist could experience. And I'm very hopeful that the agreement with Ionis to move this forward into clinical testing will be a transformational step for patients and families with Pelizaeus-Merzbacher disease. And so I will end there and hope that I've you know, story that really started with a Can you still hear me? Okay, I got a pop-up that said Zoom quit unexpectedly. Apparently not.
Can see you.
All right, great. I hope I've just provided inspiration that patients, families, scientists, clinicians, all working together really can make a difference. And even for an ultra rare disease with no therapeutics available, with our current technologies that Mitch described very eloquently, there's a lot. We're in a new era for rare diseases, and I think there's a lot of hope on the horizon to really begin to leverage some of these technologies to make a difference for diseases across the spectrum. And so with that, I will stop sharing and introduce our next speaker. So the next speaker is going to be Dr. Diana Whitmore. Dr. Whitmore is Vice President of Therapeutics Development and the Director of the Therapeutics Development Center at the Harrington Discovery Institute here in Cleveland.
All right. Thank you so much, Paul, for that kind introduction. And I'm just going to, I think the person who's sharing currently needs to stop sharing before I can share my slides. It's really a pleasure to be here. I'm very excited to be together on the agenda with colleague Mitch Drum from some years ago when we were both working in cystic fibrosis, and Paul as someone that I'm very aware of, and I've worked with one of his colleagues in the past as well. So it's a small world that we're in, and we're very, very happy to have all of you together with us here today. I'm going to shift gears just a little bit. Rather than talk in depth about a specific disease, I'm going to address that question that both of our speakers have mentioned, which is how do we, with 7,000 to 10,000 known rare diseases, tackle the breadth of that problem? And the other thing our speakers so far have illustrated in a really terrific way is the creativity and amazing new technology that's being developed every day in academic labs. So there was a time when we might have thought in the drug discovery industry that industry's job is to make drugs and academics' jobs are to advance science. And I think what's really, really awesome about where we are today is that more and more, these two different realms are working together. And we see a lot of collaboration. And we also see a lot of wonderful new technologies being licensed to pharmaceutical companies. So Paul's illustrated very nicely that kind of collaboration and how it really serves to translate the work that's happening at the bench and helps to advance those projects into actual preclinical candidates being tested in the clinic, and then ultimately moving to be approved and available to patients. And so can I just make sure that you all can see my screen? Yes, getting nods. All right.
Yes, you can.
Terrific. Okay, thanks very much. I am going to describe to you the Harrington Discovery Institute and how, with our mission, we hope to address this exact question of the great need and the depth and breadth of the science that's out there in academia, and how do we bring those ideas and those technologies into very practical, applied new drug discovery into the clinic and ultimately to the FDA for approval. The mission at the Harrington Discovery Institute is something that we think about and something that permeates everything that we do every day. And really, it is to address that need to close the gaps between discoveries of potential breakthrough therapies that are at the early stages in academia, and how to help facilitate the translation of those to ultimately candidates in the clinic and approved new drugs. And so we attempt to think about where the gaps are and how can we help close them. The Harrington Discovery Institute was founded in 2012 with a generous donation from the Harrington family. And over the next year, we've launched 11 of our first drug discovery projects. Our institute is organized around 4 key centers, each designed to address specific areas where there's great need for new medicines. The flagship program that we started with was the Physician Scientist program called Scholar Innovator. And so our first center was the Physician Scientist Center. We fund 10 or more discovery projects every year with that center, many of which are focused on rare diseases. Physicians are treating children and adults with rare diseases, and they often have really interesting insights on what is needed in terms of new therapies to address those problems. Our program for physician scientists is open to physicians who are working anywhere in the United States and in Canada, and physicians who also do research and might have some of these breakthrough ideas. Focusing in a little bit on rare diseases, in 2017, we began a strategic alliance with the pharma company, Takeda Pharmaceutical Company. And together with Takeda, we initiated the center with a call for applications across the United States in rare diseases. Today, our Rare Disease Center also includes a new collaboration with Oxford University. And last year, we also launched a new charity to mirror what we do in North America in the United Kingdom. That charity is called Fund for Cures. So let me dive in and share a little bit about how we work at the Harrington Discovery Institute. As I've already mentioned, our goal is to accelerate breakthrough discoveries into medicines that are available to patients. In today's world, the cost to develop a new drug can be more than $1 billion. So how can we possibly do this and address the many different needs for new medicines that we have? This is not something that academic grants can carry all the way through to the end necessarily, if it's going to take that much money. And often there are special challenges of creating the kinds of preclinical animal or in vitro models. Mitch mentioned those, and Paul has shown you some really wonderful illustrations of how important it is to have these animal models. But all of these things add to the cost of developing that drug, as well as the cost of operating multi-center trials throughout a country in order to enroll patients and test the drugs to make sure they're safe and that they're actually effective. And so if we think about the work that goes on, the breakthrough scientific excellent work with really novel activity happening in academia, where's the gap between that terrific work and the very late stage clinical studies that are going to lead to new drugs being approved and available for physicians to write scripts for their patients? So we consider that gap to be a combination of money, but also really importantly, it's know-how, specialty know-how on how to move things through preclinical steps and through clinical trials. And so this is the kind of gap that we are setting ourselves to help to close in the areas of where we really need new medicines, and in particular in rare disease areas. And our model, the Harrington Project model, is to look at those connections between nonprofit and for-profit contributions to the development of new drugs. So we start with the Discovery Institute on the nonprofit side of the equation by identifying these exciting new discoveries, both nationally and internationally, through a rigorous grant application and review process. Once they're selected, we try to close the gap of knowledge. We help those investigators advance their programs through those critical preclinical models and through the safety and manufacturing requirements that the FDA would require in order to allow new studies into the clinic. And so we help close that gap, importantly, by contributions from part of our network, who are the Therapeutics Development Center. And this network includes individuals who've spent their entire careers working in industry, in big pharma and in biotech, doing specialized drug discovery and development research. But they're not working for industry anymore. And so they work with us at the Harrington Discovery Institute and provide their know-how to the scholars that we select. Our ultimate goal is to help those investigators and the institutes that they're doing their scientific research in to find the right commercial partners who will make a commitment to invest in those discoveries and pay for and apply their knowledge to the final steps of clinical studies and registration for a new drug. Once a scholar and his or her project has been selected in one of our programs, they can access many forms of support from our development center, in addition to the funding. The hands-on development expertise in different areas that are needed. We also help by managing critical experiments at what's called contract research organizations. Oftentimes, if an academic researcher has the need for doing a specific animal experiment, and it's an animal experiment that they don't know how to do in their lab, we can actually go to a company whose special area is to conduct such experiments. But it takes a while sometimes to set up the relationship with that company, and it takes a while and it takes specific business know-how to manage the relationship. So we can do that for the scholars so that they can continue to do the critical science work. We also provide support in the form of coaching and strategy development for finding the best commercial partner for that potential new therapy. Looking at the heart of what we do, it's really these individuals who are represented on this circle. So imagine that this is your project that we're helping to support through the Harrington Discovery Institute. And you might have a new drug that would potentially help someone with a neurological disease. But it's a pretty early stage and the drug needs to be improved before we could take it to the next step. We might be able, through the Therapeutics Development Center, to advance that to the right clinical candidate with help from our set of medicinal chemists. And the person you might be getting your advice from could be the former head of chemistry for one of the big pharmaceutical companies. That person has dedicated themselves to sharing their knowledge to bring new programs forward. Once your therapy is now ready to go into the animal model studies, we might need to formulate it in a certain way. That means we might need to add other things to the drug, or we have to think about, is it an injectable or is it something that would be inhaled? And how do you need to prepare that in order to give it to the animal in order to do the experiment? So we can provide help there. And we can also provide help with preclinical experimental planning and clinical planning so that you're doing the right experiments to tell the FDA that you're ready to go into humans when you're ready. So each project that we have in our programs has a specific customized set of these individuals assigned to the project to help. And that is all part of the support that the Harrington Discovery Institute provides to the projects that are selected in our different programs. In the 10 years since we started, the Harrington team and our Scientific Advisory Board have reviewed over 3,000 applications, and we've selected 149 projects to support. In the next week or so, we'll be announcing a new set of award winners from our Scholar Innovator program, so that number's about to go up. We measure our success in a number of different ways. We look at the number of funded companies. As Dr. Teasar has shown us today, it's really important to think about how to commercialize your advances. And once you do form a company, you can start to collaborate with bigger companies. You can start to pull in the type of investment from venture capitalists and others that will be required to support the cost of the future studies. We also measure our success by the number of candidates that have entered the clinic through the projects that we've supported. And so far, 17 of our projects have entered clinical studies. One of our exciting rare disease projects is for a pediatric cancer, and it's in phase 3. So we're really looking forward to the day when we might be able to have a brand new drug on the market through one of the investigators that we helped with our programs. And finally, we also look at how many of the projects are licensed by big pharmaceutical companies in order for them to move those forward to the clinical studies. Importantly, although we're headquartered right here in Cleveland, we look for the best breakthrough science across North America and the United Kingdom. With 7,000 plus rare diseases in need of new medicines, we have to really throw that net wide and look for the best and most creative breakthrough therapies as broadly as we can. We also need to build our network of investigators because of how important it is for folks to collaborate. Our previous speakers have already illustrated the power of bringing different scientific minds together. And we do have an annual symposium at which our scholars would come together and share their different research results with each other. To give you an idea of our ambitions for impacting the rare disease field, our 2 rare disease specific programs have so far reviewed 141 applications from North America and 46 applications from the United Kingdom. And from those, we funded 24 exciting projects that we hope will yield new treatments. Our transatlantic program in the UK is based on the same model that we've had success with here in the US. Just last year, we launched that program and selected the first 5 projects through our UK charity Fund for Cures. And we'll be sending out the second call for applications later on this year. The Oxford Harrington Rare Disease Center combines Harrington's expertise with the scientific excellence of the world's leading university. The center is focusing its efforts on rare disease research in 3 main areas: neurological, developmental, and pediatric oncology. Together, we plan to put 20 new drug candidates into the clinical development in the next 10 years. So that's ambitious, but we're dedicated to getting that done. Through our partnership with Oxford University, we can also give any of our rare disease scholars access to Oxford's world-class facilities. Whether our scholars are located in the US, Canada, or the UK, all benefit from our approach to sharing know-how from our Therapeutics Development Center. That's our group of pharma experienced experts, and where they have their greatest impact is hands-on. And so if you imagine that you are one of those scholars and think of representation as this little red figure in the middle of the circle, this is where all the specialized, customized input could come from medicinal chemistry, from formulation, from business development, and also from regulatory. And so we think of ourselves as being an extension of the team, of the lab team that we're working with. And we invite those scholars to think of us as an extended member of their team. We do not charge the scholars. It does not come out of their grant funds for all of this support that we provide. And because we all have our own scientific backgrounds, we all get extremely motivated and very excited when we see that progress. That gives us encouragement. So here are some of the faces and projects that illustrate what we've been privileged to do. 2 of the projects we funded in 2018 have now moved to commercial partnership. Dr. Justin Nachita has a treatment for ALS, otherwise known as Lou Gehrig's disease, and he's spun it into a new biotech called Acuristem, which now has this potential new drug at the preclinical stage. Dr. Ed Grabczyk is at Louisiana State University, and his novel discovery was to use our own DNA repair system to halt progression of Huntington's disease and Friedreich's ataxia. This is a type of therapy that Mitch and Paul have both mentioned that we call an ASO, or antisense oligo. That program has now been licensed or optioned to license by Takeda Pharmaceutical Company. We're also very, very excited about the work that Dr. Lee, Jeannie Lee, has published with her treatment that could be extremely impactful for girls with Rett syndrome. Today, there isn't a therapy that addresses the disease, and so we hope that this therapy will be into the clinic sometime in the next couple of years. And finally, Dr. Jim Shaman at the University of Michigan has a new oral therapy for a rare form of neurological Gaucher disease, and he is currently assembling the toxicology package for a preclinical candidate. So I want to thank you so much for this opportunity to share some of the Harrington Institute's work in rare disease drug discovery. We're very proud to be part of this community, and we take our inspiration from all of you. You can learn more about us at another webinar coming up next Monday. And you can also learn more about our institute on our website, or you could follow us on Twitter. So that's the end of my presentation. Thank you so much for your attention and for the opportunity to tell you about us. I would like now to introduce the next speaker. He will be Jeff Codman, who's going to share some key insights from the patient perspective. And I'll just stop sharing my screen if I can.
Good morning. Thank you for the opportunity to recount our experiences with rare disease and conditions to provide support to those who are dealing with their own unique situations. 17 years ago, my wife got some of my close friends together for my 40th birthday. 3 of our children were upstairs in bed for the night. One of our sons, a few days from his 4th birthday, was sick and laying on the couch in our family room. We checked on him periodically during the evening. His condition never improved. He had been weak and lethargic for most of the previous week. This was not normal for him. We had contacted the pediatric practice multiple times that week, and we were reassured each time that if he didn't have a fever, he was probably not that sick. He never had a fever. As our party concluded, my wife decided to take him to the ER. In the ER, they put an IV in his arm to rehydrate him. Within minutes of the start of that IV, he suddenly manifested a set of unusual symptoms. His gums became bright red. His tongue looked like the surface of a strawberry. The tips of his fingers and toes started to shed skin. They told my wife he had the classic signs of Kawasaki disease. Luckily, we were extraordinarily blessed by the fact that the head of pediatrics at the hospital at the time was a doctor who knew more than most about Kawasaki disease. He, in fact, had worked with Dr. Kawasaki. That was incredibly reassuring. But our son was seriously ill. We were told that a dose of IVIG would turn off the inflammatory response and reduce the chance of permanent damage. After the first dose, his condition did not dramatically improve. I remember holding him in my arms and he could barely find the strength to move. The doctor decided to give him a second dose while telling us that only one child in all of his years of treating patients with Kawasaki disease has ever needed a second dose. We were lucky to see our son did in fact improve somewhat after that second dose. We were allowed to take him home after that week that he had spent in the hospital. However, a week later, we brought him back to the hospital because the symptoms had reappeared. A third dose of IVIG was, in fact, the final solution to this nightmare. 18 months after our son's illness, I found myself with a sinus infection that lasted over 8 weeks. I was freezing, I lost 20 pounds, I couldn't get the sound of my heartbeat and the blood flowing in my ears. After 4 different doctors, a blood test was finally performed, and I was subsequently admitted to the hospital in complete kidney failure. They did a kidney biopsy and told me that my kidneys were 89% destroyed and that the cause was a vasculitis called MPA. The doctor gave me high-dose steroids and Cytoxan. I responded remarkably well to this older protocol. In 2011, I ended up having a hemorrhagic stroke caused by an AVM. In the ER, they gave me Dilantin as a prophylactic to prevent seizures. After I went home, about 3 days later, my mouth tasted like iron, and my daughter came into my office and she saw that I had petechia on my arm. I was having an adverse reaction to the Dilantin, which killed off all my platelets. Stopping the medication and multiple transfusions later resolved that. 6 months later, I had the brain surgery to remove the AVM, followed by physical therapy to learn how to walk again. But later that same year, the vasculitis flared again and sent me back into the hospital. The immunosuppressants were not doing the job anymore following that stressful year. So I was switched to the monoclonal antibody Rituxan. Today, if you met me or my son, you would not have any indication that any of this had transpired. Rare diseases, rare conditions, rare treatment protocols, rare adverse reactions, and rare outcomes. All of these have become our new normal. Knowing that there are other people out there that have experiences like yours can give many of us the confirmation needed for us to maintain a sense of normality. This gives us the strength to move forward in our daily lives with hope for tomorrow. Now I'd like to take the opportunity to introduce our next speaker, Tracy Greenberg, who is a patient advocate. Thank you.
Thank you very much. And I wanted to thank everyone here today, for all the presenters and everyone who is involved in the planning of this event. Everything's set up and you could hear me and see me well. And essentially, I'm here to represent the rare disease patient community. I'm an advocate for rare diseases as well as accessibility for those with needs for removal of barriers. I serve as chair on my city's ADA task force, and I also serve on Metro Health's Patient Family Advisory Board. And I've been working with the Rare Disease Day Planning Committee for several years. And in the picture that you're looking at right now, I was actually part of what was the first Rare Disease Day event at Metro Health, which was basically me with a table sitting outside the cafeteria and flagging down the providers that were walking by to make them aware of rare diseases and the need for the attention for change and progress in this field. So myself, I do have a rare disease. I have one of these single gene rare conditions that Mitch was talking about at the beginning. I have a disease called mucopolysaccharidosis, MPS for short, and there are several types. The type that I have is type 4A, and that's called Morquio syndrome. And essentially, I'm missing an enzyme, which caused all kinds of problems when I was growing with the development, primarily skeletal, and also it does affect other systems on many levels of the spectrum. It does affect respiratory and some neurological effects as well because of instability in the C-spine and other joint and musculoskeletal instability. So I didn't actually know that I had Morquio syndrome until age 42, and I'm 47 now. I was first diagnosed with, my brother and I both had evidence of something, and I was first diagnosed with bone dysplasia in the 70s. And then in later on, they said we had multiple epiphyseal dysplasia. And then when I was in my 20s, I saw a genetic specialist who suggested that I had spondyloepiphyseal dysplasia. And all those things are resulting from Morquio syndrome. So those are all conditions on their own. But so I was basically underdiagnosed. And at age 42, I started really looking into, can I get tested to find out exactly what I have? And I joined the team over at Akron Children's, and they helped me come to the conclusion that I had Morquio syndrome, and I was tested, and my brother and I both do have that. Both of my parents were carriers of it as a recessive trait, so they aren't affected, but 2 out of 3 of us children were. And so I'm here to talk about a couple things. I'm going to talk about transitioning from being a pediatric patient into the adult world, because there's a lot of barriers there. Now, when you have a rare disease such as mine that's a developmental disability, a lot of times it's better to be in the peds world, because the providers can understand better the developmental situation and see that, well, maybe perhaps these are bones that didn't fuse, as opposed to you're missing bone and you have bone loss and these are broken bones, which is a lot of times the difference in information that I get from the different adult versus peds world. I do have a PM&R doctor who is a peds doctor, and I'm planning to go to the Cleveland Clinic for surgery. And I asked her, can you be my quarterback for my recovery? Because they don't really have a floor that I can recover in for comprehensive care. And she said, well, I can't really I can provide you care, but in the hospital setting, I can't cross over into that adult world. So I'm going to have to refer you to another doctor. And that's a big hardship for me, because now I have to kind of bring my new doctor up to speed, as opposed to just being able to kind of have my current providers who also have the capability to provide for adults, to just kind of transition into the care for me as I get older. So something really needs to be done about that barrier between the adults and the pediatric world. If we are lucky enough to live through a childhood rare disease into adulthood, which is ultimately the goal of all of this science, then we want to be able to treat the adult patients that are living through the childhood disease, and that's a big gap. So that's my 2 cents on that. So I'm here for the long haul to advocate for that. And then I just have one other thing I wanted to talk about. And I just want to point out the importance of art in medicine. I'm a singer-songwriter, and I've learned how to play guitar, which is, I've always known that was kind of extraordinary and above and beyond, because even though I didn't know that I had Morquio syndrome, and I knew that my joints were affected, and so playing guitar to me kind of felt like a miracle that I could do that. So I was able to overcome in that way. And it was a challenge to keep up, but it's a therapy, and I have made a profession out of it. I've released 6 albums, and I perform professionally in Cleveland, Ohio. And I've made a name for myself in the not only the rare disease world, but also in the music world, for advocating for musicians that also have conditions that affect their mobility and ability to access the stage. And that organization is called RAMPed. I could put that in the chat after my presentation. The website to access for that is ramped.com, ramped.org. rampd.org. So I just want to talk about the art and medicine for a second. I made this mug in ceramics class, and it, helped me get my mind off my pain. It helped me, use my arms and stay moving. When you're in that art zone, it takes away from the challenges that someone is facing when they have a rare disease or any type of illness. So with that being said, I hope that everyone will check out my music at tracymarie.com. And thank you so much for all your great work, and I hope to connect with everybody in the near future as well. And I'd like to pass it on to our next presenter, Dr. Mary Rensel. Thank you so much, and I appreciate your ears.
Tracy, amazing. We got a chat. I love the art. And hi, everybody. I'm Dr. Mary Rensel. I'm going to start sharing my screen. Yeah, that's powerful, Tracy. I appreciate it. And just as a little segment, there are some national organizations looking at that transition. Just so we should chat, and I could walk you through what they've been working on, because it's a huge issue and people do know it's very important. And yeah, I would love to. I'll find you, Tracy. We'll chat. Okay, so I'm going to speak about, the collective of days like this, of us, getting together and putting our heads together and hearing the impactful, patient perspective and the research perspective. And I'm a clinician. I'm a neuroimmunologist at the Cleveland Clinic Mellon Center, the center that treats multiple sclerosis. I treat both pediatrics and adults. I also treat NMOSD, and that brings me here today. I want to share with you just what it's like in the clinic. Let's see, get going here. There we go. There was a day when I would, share the diagnosis of NMOSD with my patients, and there were no websites, there was no one to go to. So I just had a patient yesterday who was diagnosed when she was 13. And, she was telling me what it was like when she was 13. She's now in her late 20s and she's working full time and living with some disability, but living well. And she said, oh, it would have been so nice to have some organizations that would have helped us to be able to get together and share resources, et cetera. So, just know that this group, I really appreciate Nord and all the involved groups and the researchers sharing their knowledge, because just people looking back to when they were diagnosed decades ago or 15 years ago, there wasn't that infrastructure in order to find all the resources. So when we put our heads together like this and put our hearts together, this collective can happen where we can support each other. The diagnostic journey can be difficult, like we've heard from the patient perspective. Sometimes we see a symptom of the disease and it's, just like with Tracy with the bones, and that's it. And we're not thinking kind of a wider view, what would be causing that issue. As a neurologist, I tend to focus on the neurologic issues. Cardiologists would focus on the cardiac issues. And so, having these kind of days where we can put our heads and minds and hearts together and make the diagnostic journey more clear and more supportive for the patients is incredibly important. So we're going to talk about that and what is shared decision making in the office. We're going to talk about that, and then coping for both, those living and supporting somebody with a rare disease and those working in healthcare. So what about that diagnostic journey? I can speak to NMOSD. That's a rare disease that I see regularly in the clinic. And there's a unique urgency to that, to giving that disease the correct name and treatment plan, because the relapses or abrupt symptoms that can happen from that disease can be life-changing and or fatal. So we need the correct name and a treatment plan as soon as possible. And if you speak to 10 people with NMOSD, everybody has had their own journey on when and how they were told the name of the disease and how long that took. So when we finally do give the correct diagnosis for that, I then work with the patient and their caregivers and supporters. This is what you need to say if you go to emergency room. Like, don't say NMOSD because people won't know what they're talking about. And I've been checking with patients, like, do the emergency rooms know that name? Emergency room doctors know that name? And they said, no, they don't usually know. But it's been interesting. I've had a few patients recently share with me that if they went to emergency room, let's say for COVID or another reason, the emergency room docs, a few of them have said, tell me about that disease. What does it mean? So there's been some curiosity, which I think is a great place to start when we don't understand something. We want to try to feel the empathy and get to know somebody. It's just simply to start with our communication with with curiosity. What's it like for you? What's that disease like to live with? What medicines you have to take? What do you need to do? How many doctors do you see? So it was really neat. So when an ER doc sat with one of my patients recently, I was like, tell me about that disease. And they felt really heard at that moment. But because of the rare diseases, a lot of times I instruct my patients, like, these are who's on call at night. This is the number to call. You need to call with X, Y, and Z symptoms. Don't give it any more than this many minutes or this many hours, because this disease does have an urgency to it. Not all diseases do, but NMOSD definitely does. So I try to be very clear in the communication, when to call me, when to call the nurse on call, when to call the doc on call, and when to go right to the emergency room and don't waste any time, and what to say in the emergency room. So that's the challenge with rare diseases, is that in medical school, we learn a lot about the common diseases. Sometimes we learn a little about rare diseases, and it depends on what your specialty is and how much you know about rare diseases. So a lot of times the patients are teaching us, in the emergency room and variety of settings. And then the power of the advocates. So patients say, can my, outside of COVID, we've done a lot of FaceTime and office visits because, there was a time people couldn't come in to the office visit, a caregiver or supporter. I love when people bring supporters in their room in our office visit with them. I am always glad to have an extra set of ears so we can hear the treatment plan and they can share their observation, how things are going. I think it's going better, it's worse. What are the latest challenges? So I'm a huge fan of advocates as a caregiver, as a friend, as a supporter, as a partner, and then also to tap into some of these resources like today, to tap into your state's resources and national resources. So we think very, nuclear at the beginning, just who do you have in your house or that would come and be able to help you, but then who is in your region, that if there's associations or there's meetings or these webinars, and then, state and national. So build a network of support and advocates and get to know them, because people, the passion of advocates is unstoppable, right? So days like this, now we're going to have all of these days and all these NORD days throughout the country, and people will share their passion of what they've done and how they've gone to talk to their, the government, the state government, the national government to make change. And that is incredibly powerful. The other power comes from within our office visit, knowing what's important to me and for you to know what's important in our treatment plan. So again, we start with that curiosity. So I want to know what's most important to you in our treatment plan, and I share with you what's most important to me. If I had this disease and as your treating doc, this is most important to me, how well it works and how safe it is. Those are generally my two leading principles, but I want to know what's your leading principle. And that's the definition of the shared decision making. So I want to know what's important to you, and I want you to know what's important to me, so that we know each other's values to make this decision together, and we set goals. And I tell you, this treatment, this is my goal for this treatment. And it may be a really high bar, but this is our goal. And this is why I want you to call me along the way or reach out if you're having troubles, because we're trying to hit this goal together. So we try to make that very clear. And this is a kind of a relatively new concept in the office, in the shared office visit, to have this shared decision making between the patient and the provider. So it's very clear what's important to you. I generally start the visits with, what do you hope to accomplish for this visit today? And so that and people say, oh, I put it on, now everyone puts it on their phone. They used to bring notes, but it's always on their phone. And I say, okay, before everybody leaves, check your list on your phone. Did we hit everything? So we want to make sure I'm addressing what's very important to you, and I'm sharing with you what is important to me as far as treating the disease as best as we can. And coping, this, when we're talking about a chronic disease, the best way I've heard it explained, and this was a father explaining what it was like when his son was born with autism. And he said, it was like we had planned for a trip going to Belgium. And when we got there, we actually ended up in, Italy. And I was ready for a trip, and I knew I was going on a trip, but I didn't know I was going on this trip. And I think for me, that really helps me understand what it's like to adjusting to a new reality. So when we talk about coping, the the thing that our neurobiology needs most primarily is belonging. So groups like this, because you all understand rare diseases, this is a wonderful community to feel that this community understands what it's like to either live or research or care for people living with a rare disease. So the first step in our neurobiology, what we really need before we can really live our best selves, is we need to feel like we belong somewhere. And that's so challenging with a rare disease, because a lot of people I diagnosed with NMOSD, they don't have anyone in their church or their temple or their mosque or their school or their town, sometimes even their state that they know with NMOSD. So a rare disease, that belonging is taking away. And that's why I'm saying these communities that have been, have been flourishing over the years and have built by all of your hard work, they really matter, because as humans, that's the primary thing we want, is to belong. And so we need to fulfill that need, and this kind of group can do that. And if we feel like we belong, then we can start to trust our caregivers. We can start to trust those in the community. So that's our neurobiologic need. And then we can get to the creative level and really try to help people. So this is when you think like, I don't know if I'm coping with this disease. No one understands me. It's a rare disease. No one I know has it, or no one understands. I go to the emergency room, no one's ever heard of it. Then, maybe get your sustenance or, from the group that does understand you, and then bring that out and educate people, because, there are some people who are actually interested and curious and want to learn more about it, want to learn how to help and make sure they don't miss this diagnosis in anyone else. And so working on belonging and finding a crew, finding people around you and putting people around you that are really working on something that's important to you is number one. And this goes for those living with rare diseases, caring for those with rare diseases, and those of us working in research and healthcare. And then trusting, that's hard to get to the trust level with new people. But if we see that the values are true and that we agree, we really want to work together to end these diseases, like these amazing talks today from the researchers. It's so, very powerful and positive, and it's really wonderful to hear all the good news. So it's exciting, all the creativity that's going on. And then as humans, then we can get to the flourish level, right? So if you find someone, or you feel like yourself that you're not sure you're doing okay, always reach out to a professional. The talking kind of supportive doctors are counselors, psychologists, social workers. Those are mental health care teams. I would for sure have that at the beginning, especially with that adjustment period when you went on a trip and you got to a destination you didn't know you were going to get to. There's a lot of professionals that can help people adjust, and then find a team that you feel like you belong to, and people you can trust. And then you'll find you can then flourish and get to that creative zone and really live a life that's very interesting, like Tracy telling us about her music and everything else. So today we talked about the diagnostic journey. It's a journey, and it's sometimes very long for folks. Sometimes right away it's clear what it is, but a lot of people have had really challenging and I would say disappointing. You want to get a name and a plan right away. And so the diagnostic journey can be a challenge. The shared decision-making is an important concept. If you don't feel that with your provider, I would suggest that you bring a list of 3 things. This is what I want to accomplish in my visit today. This is what's important to me in my treatment plan, and make it very obvious and clear to them so that they know exactly what's important to you. And if you're not feeling heard, I would keep, going back to those lists. And then the coping, the first thing is of course, the belonging. So if you're not feeling like you found a team or a crew that understands what you're going through, keep searching, because there's people who are very passionate about supporting others, and you will find them if you keep looking, like Nord, et cetera. So I love this quote by Samantha Power, but it says, all advocacy is, at its core, an exercise in empathy. Thank you for your attention. I want to let you know about a day we're having on March 30th. It's sponsored by Connor B. Judge. Thank you. And we are going to update on the latest on neuromyelitis optica spectrum disorders for pediatric and adults. It's there's no charge for this day. It's all virtual. So you can just get the link there and sign up. And we would love to see you. It's going to focus on pediatric and adult NMOSD, those living with it and those caring for or just want to know something more about it. So we'd love to see you on that day. And reach out. I'd love to connect with you all. Thank you for your attention. Oops. Okay, I'm supposed to introduce Megan. So she has a video for us. She's busy studying at college, so here she is.
Hi, my name is Megan Wolf. I'm a film editor from Cleveland, Ohio, and I'm about to graduate from Ohio University this semester, majoring in integrated media with a minor in film and a certificate in diversity studies. And while this career aspect of my life is beginning to take off, I've also really had to focus throughout my high school years and going into my college years, now going into my future, with neuromyelitis optica spectrum disorder. I was diagnosed at the age of 15, so an incredibly young age, and directly during a time where I was figuring out who I was and how different labels can be attached to certain people depending on certain aspects or events in their life. And that's one of the things that I wanted to talk about today. Being 21 now, I have attempted to make my life as seemingly normal as possible. You know, I have an invisible illness, so there are aspects of my life that I can kind of cover up. And I can share this reality with the people that I trust, but I also have that aspect of saving who I am. And I noticed that being in LA last semester for a domestic abroad program, that I was really doing that in order to kind of save face and seem like I wasn't weak. I was so afraid of judgment that I would receive for having a chronic illness that it became a shame that I had on myself. And that brought up an interesting conversation that I had almost with myself, just wondering, why am I trying to seem normal? Because what is normal? You know, trying to fit in it with everybody else. And then it really made me start to think about my own experience. How there's some people that look at me and they say I'm so strong, I'm inspirational. And then there's other people that look at me and think I'm weak, I'm delicate, I need to be coddled because there's something wrong with me. And there's no middle ground where I'm just a human being. I'm just living my life. We didn't have a choice going into all of this medical stuff. We had to be strong and we had to just deal with it in order to survive. In a society that continues to say we are working towards representation, why is this still such an enormous problem where we cannot see ourselves as human beings and instead put labels on each person and kind of pick and choose who we want to associate with by how comfortable we feel? I feel like I had to conform to this box of how I was supposed to look and feel. And because I am so young, I felt like I just couldn't relate to fellow peers around me because they didn't know how to have that difficult conversation when it was necessary. So it created this distance, and I didn't know how to get myself out of that box because I felt like I was so tied down to this expectation of how I was supposed to be. In this way, I pushed down trauma, and this led to deeper and deeper anxieties about life and socializing. It wasn't until the pandemic in 2021 where I really realized, oh my God, you've been pushing down all of this anxiety, all of this trauma for so long, and you never addressed it because you felt that all you had to do was just pretend that it didn't exist in order to be accepted. And my anxieties, my experiences, they're all valid. And that's how that should be for everyone in this community. I decided in 2021 I was going to work harder to be more authentically me and to understand that those people that don't accept me are not people I want to have in my life. But it's about finding those people that do understand and care. I realized by not letting that tie me down, by not letting my anxieties get the better in me. I was able to overcome some of those fears about those labels in order to find out what I want to do in my life, why I'm living my life, why I'm continuing to survive. What does that all mean? And I finally feel like in 2022, I'm beginning to recognize that and grow from the pain that I've experienced through this period of having NMOSD. Hi, my name is Megan Wolf.
Bear with me, everyone. Chelsea has a thank you and a video for us as well that I will share now.
Thank you to everyone who participated and attended the 3rd Annual Ohio Rare Disease Day. While diseases, including neuromyelitis optica, which my brother lives with, are rare, there is a powerful strength in numbers in the rare disease community. Together, we can move mountains, including bringing awareness to rare diseases, raising funds for research to find more effective treatments and ultimately cures, and working to implement government and societal policies that protect and improve the lives of rare disease patients and their families. What, like it's hard? The rare disease community has done it many times over, with fighting for the Orphan Drug Act to spur incentives for rare disease research and pushing for fairer drug prices. We will keep on the fight. The Ohio Rare Disease Day is a shining example of collaboration. We at the Connor B. Judge Foundation, like many other family and community-based nonprofits, are small but mighty. Our strength lies in our network and joint mission with other like-minded groups. We are forever grateful to NORD for coordinating Ohio Rare Disease Day with us, as well as the combined resources and efforts of the Gus B. Jackson Foundation for NMO. As all patients and their family and friends know all too well, the compassion and drive of clinicians and scientists change lives for the better. It is collaboration and commitment to overcoming the hardships of rare diseases that makes our partnerships with clinicians, researchers, and administrators from the Cleveland Clinic, Case Western Reserve University, Metro Health, and university hospitals that makes us powerful. My family and I know deeply that rare diseases rock your world. And we know even more that there is optimism, light, and love within the rare disease patient advocacy community. For that, we are grateful to each and every one of you. We are in this together. Thank you, and I look forward to all that we achieve together in the next year for those impacted by rare diseases.
Okay, so this brings us back to our wrap up and what you can do. Please join us at Ohio RAN. You can follow us on Facebook. We'll also drop a link in the chat for our Ohio page. And sharing your story, as we've shown today, can be an incredibly powerful way to advocate. I want to take a minute to say thank you to all of our speakers, to Pam from the Connor B. Judge Foundation for helping to plan today, to NORD and all of our sponsors for their work in organizing our event and helping us kick off Rare Disease Day in style in Ohio this year. And thank you all for joining us and for making it great. If you're in Cleveland, look out for Tower City and Rock and Roll Hall of Fame to be lit up today to celebrate Rare. Our door prize winners are Sheila and BMW 32. If you want to contact us with the email, again, I'll drop it in the chat. And we hope you will continue to join us in our efforts to make Ohio a great place to live with rare disease. Thank you so much, everyone, for joining us today.
I just want to add to the winners, we need your mailing and shipping address. So if you can email us with your actual shipping address, that would be great.
Perfect. Thank you, Kristen.